Mutations. Yeah, the last chapter was genetics. Now this is also related to genetics. When this mutations are permanent changes in the DNA sequence of a gene. When there is a permanent change in the DNA sequence, that is known as the mutation. These changes can affect how genes function, potentially altering the characteristic of an organism. Mutation can occur in any cell of the body and may be passed on the next generation if they occur in reproductive cells. Germline mutations. So if they occur in the reproductive cells, for example in the sperm or egg cell, then they can be transferred to another organism as well. Causes of mutation: spontaneous mutation, and then we got induced mutation. Spontaneous mutation: these occur naturally and randomly during DNA replication. Errors can happen when cells copy their DNA before dividing, leading to small changes in the genetic code. Although cells have repair mechanism to fix errors, sometimes cell can fix the error. Some mistakes persist and become mutation. Some mistakes cannot be fixed, and then it becomes mutation. Induced mutation: these results from exposure to environmental factors such as radiation. Radiation is the first cause, like UV rays from the sun, X-rays, or nuclear radiation can damage DNA and lead to mutation. Or chemical, let's suppose taking tobacco smoke, pesticides, or industrial pollutant may alter DNA structure or viruses, etc. Effects of mutation: the first one is harmful mutation, second one is beneficial mutation, and the third one is the neutral mutation. Some mutation cause genetic disorder or disease by disrupting normal protein function. For example, sickle cell anemia caused by a single DNA change in the hemoglobin gene. Like even a very small minute change can cause mutation. Cystic fibrosis resulting from a mutation in the SFTR gene, and cancer, which can develop when mutation affects genes controlling cells growth and division. Beneficial mutation: in some cases, mutation provide an advantage that enhances survival and adaptation. For example, lactose tolerance in some human population allows adult to digest milk. Antibiotic resistance in bacteria help them survive exposure to antibiotics. And color vision: variation in certain animals improve their ability to find food or avoid predators. So these are all good mutation, beneficial mutation. And the last one is neutral mutation. Many mutations do not significantly affect an organism's function or survival. These mutations might occur in non-coding region of DNA and result in minor changes that do not impact protein function. Original DNA, for example, look at the original sequence and look at the point mutation. If you look at here, there's T over here, and here is C. So here there was thymine, and here there is cytosine, which is the mutation. Types of mutation and their effect. The first one is based on DNA sequence changes, and in that, then the first one is point mutation. What is point mutation? These involve a change in a single nucleotide building block of DNA. For example, there are three types of it. These are very important: substitution, insertion, and deletion, and they are super easy. Substitution: if you look at here in this diagram, the normal one, ATG, CGA, TTC. And now if you look at here, ATG, TGA. So this middle one has been substituted. The CGA has been substituted by TGA. Now, so this is known as substitution, while insertion is, if you look at here, ATG, CGA, TTC, and here there is like one A added extra. So this is known as insertion, something which is inserted. And deletion, if you look at here, there is ATG, CGA, TTC, and there is AT, and there is GA over here, and here CGA. That is the only difference. So there is like a slight difference in the gene sequencing, which can affect the whole cell. Frame shift mutation: these occur due to insertion or deletion that shift the reading frame of the genetic coding, altering every amino acid after the mutation. For example, Tay-Sachs disease is caused by a frame shift mutation that disrupts an enzyme involved in lipid metabolism. Based on effect on gene function, silent mutations. A mutation that changes a DNA sequence but does not alter the final protein because the genetic code is redundant. Example, a change from GAA or GAG still codes for glutamic acid, meaning there is no functional difference. And number B is missense mutation, a mutation that changes a single amino acid in a protein, potentially affecting its function. For example, in sickle cell anemia, the amino acid glutamic acid is replaced with guanine, leading to misshapen red blood cells. And the last one is nonsense mutation. So a mutation that introduces a premature stop codon. Stop codon is a codon on which the gene stops completely, causing the protein to be incompletely and likely non-functional. For example, Duchenne muscular dystrophy occur due to a nonsense mutation in the dystrophin gene, leading to a muscle degeneration. The third one is based on chromosomal and inherited mutation. Now, number one is chromosomal, number two is inherited. What is chromosomal and what is inherited? These involve large changes in the chromosome structure. First one is deletion, loss of a chromosome segment, duplication, extra copy of chromosome. So deletion is loss of chromosome, duplication is extra copy of chromosome, and insertion is a chromosome segment flips and reattaches. And the last one is translocation, a segment moved to another chromosome. Like a segment will move from one place to another, one chromosome to another chromosome. And inherited mutation, these are passed from parents or occur during an individual lifetime. So these are like mostly genetic. Germline mutation inherited through reproductive cells. Somatic mutation occur in body cells and are not inherited. Effects of mutation. Mutation can be harmful, beneficial, or neutral effect. The number one is harmful mutation, number two is beneficial mutation, and the number three is neutral mutation. Let's talk about number one. So disruption, normal gene function leading to disease or disabilities. For example, cystic fibrosis and cancer, we already studied that in the previous slide. And beneficial, lactose tolerance and antibiotic resistance in bacteria, which is a good beneficial mutation. And neutral is the silent mutation, change DNA but do not alter protein function. You can see the normal gene and the mutated gene, there is like a small difference. For example, normal protein will look like this and abnormal protein will look like this, or there will be no protein at all if mutation occur in the gene. Natural and artificial selection. Natural selection proposed by Charles Darwin in the process by which organisms with advantageous traits survive and reproduce more successfully, gradually shaping species over time. It relies on variation. Genetic differences arise through mutation, recombination, and genetic flow, creating diversity within a population. Survival advantage. Organisms with favorable traits have a better chance of surviving and passing on their gene inheritance. Beneficial traits are passed down to offspring, becoming more common over generation, leading to evolutionary adaptation. The number two is artificial selection. And artificial selection is a human-driven process where individuals with desirable traits are intentionally bred. Remember, they are intentionally bred to enhance specific characteristics. They have been widely used in agriculture and animal breeding for centuries. Selective breeding, genetic breeding. Now there are two types of breeding. Genetic engineering. Selective breeding. Humans breed organisms with favorable traits over a generation to enhance productivity, appearances, or resistance. For example, high-yield wheat varieties and disease-resistant cattle have been developed through selective breeding. So you can get, for example, let's suppose if you want something in high yield, for example, let's suppose wheat or fruit or anything, you can do selective breeding. While genetic engineering is basically scientists modify DNA directly to introduce beneficial traits, speeding up the process compared to traditional breeding. For example, the wild cabbage Brassica oleracea has been selectively breeded into Brusseli cauliflower, kale, Brussels sprouts, and cabbage, each with unique traits suited to human needs. Comparison. The features. The natural selection is natural environmental pressure, while artificial selection is human controlled. Speed. The natural selection is slow and the artificial is faster. Purpose. Survival and adaptation, and artificial is desired trait for human use. The gene basis of evolution. How mutation drive evolution. Mutation introduces genetic variation, which is essential for evolution. How mutation drive evolution? So genetic variation and adaptation and survival. For example, so mutation introduces genetic variation, which is essential for evolution, and beneficial mutation spread through population over generation, increasing adaptation and survival. For example, evolution of antibiotic resistant bacteria due to random mutation that helps them survive antibiotics. For example, mutation and evolutionary process. Natural selection, genetic drift, and gene flow. Again, natural selection, genetic drift, and gene flow. Let's talk about the first one for now. Mutation that improve survival or reproduction are favored in past one. For example, peppered moths developing darker wings in polluted areas for camouflage. And genetic drift. Random changes in mutation frequency, especially in small populations, can lead to the loss of fixation of traits. For example, founder effect in isolated island populations. Gene flow. Migration introduces new genetic variation into populations, increasing diversity and adaptability. For example, interbreeding between Neanderthal and early humans. Evidence of evolution. Fossil records show gradual changes in species over millions of years. For example, transitional fossils like Archaeopteryx link dinosaurs to modern birds. So there is this belief that these fossils were linked to the dinosaurs, or to those fossils link that dinosaurs were actually the modern birds. And comparative anatomy. Similar bone structure in different species indicate common ancestry. For example, the pentacetal limb in humans, bats, and whales. So it is same in humans, where bats and whales, which tells us that they were in one stage they were similar, but then they got evolved. And the third one is genetic evidence. DNA similarities reveals evolutionary relationship. For example, human and chimpanzees share 98% of their DNA. Evolutionary theories explains the process that drive changes in species over time. Number one theory number one is Lamrecht theory of inheritance of acquired characteristics. He proposed by Jean Lamrecht suggested that organisms develop traits based on use and disuse. For example, giraffes develop long necks by stretching to reach leaves, traits acquiring during a lifetime. So according to him, he said that giraffes have the long necks because by stretching to reach the leaves, because they were not able to reach the leaves, that's why they have the long necks. Now, Darwin's theory of natural selection, proposed by Charles Darwin, and organisms with beneficial variation survive, reproduce, and pass traits to offspring. And based on variation, competition, survival of the fittest, and inheritance. Darwin theory is the most accurate one, which we use even till now. The last one is mutation theory, proposed by Hugo de Vries. It suggested that mutations, sudden genetic changes, are the main cause of evolution. Some mutations provide advantages, leading to new species over time. New Darwinism, modern synthetic theory, combines Darwin's theory with genetics, explains evolution through mutation, natural selection, genetic drift, and gene flow. Mutation, natural selection, genetic drift, and gene flow. Alright. Is supported by molecular biology, fossil records, and genetic studies. Number 5 is punctuated equilibrium. punctuated equilibrium. Evolution occurs in rapid bursts followed by long periods of stability, contrasts with the idea of slow gradual change. As you can see here in this picture, original short-necked ancestor, then keep stretching neck to reach leaves higher up on the trees and stretching until neck becomes progressively longer. As you can see in the first one, the neck is shorter, and the last one, the neck is longer. Heredity and environment. The transmission of genetic traits from parents to offspring through DNA, it determines character. So the transmission of genetic traits from parent to offspring is known as heredity. For example, like the eye color, height, and genetic disorder. Environment. External factors like climate, nutrition, education, and lifestyle that shape development and behavior. Interaction of heredity and environment. For example, some traits, for example, blood type, or purely genetic. Others, for example, intelligence, weight, result from both heredity and environmental influence. An environmental factor can modify gene expressions. For example, Jennifer and Kevin, identical twins born with genes that absorb fats twice as fast as the average person. Jennifer eats healthy low-fat. Karen eats fatty unhealthy food. Jennifer obesity genes not triggered, while Karen obesity genes are triggered. Now, what is the role of mutation in modern research? Number one, disease research. Understanding genetic mutation is crucial for diagnosing, preventing, and treating various diseases. Genetic disorder. Mutation in specific gene can lead to inherited conditions like cystic fibrosis, sickle anemia, and Huntington's disease, which we studied earlier before. Studying these mutations help in developing treatments and early detection of method. Cancer research. Many cancers arise due to mutation in gene regulating growth cells. And the last one is gene therapy. Scientists use gene therapy to correct harmful mutations by replacing faulty genes with functional ones. For example, experimental treatments for muscular dystrophy and sickle cell anemia. Now, genetic engineering. Advancement in gene editing and modification rely on controlled mutations to improve healthy agriculture and biotechnology. CRISPR-Cas9, a revolutionary tool that allows precise editing of DNA, enables scientists to correct genetic defects, develop personalized medicine, and enhance disease resistance in crops. The last one is GMOs, genetically modified organisms. Scientists introduce beneficial mutation into plants and animals to enhance agriculture. For example, pest resistance BT cotton reduces pesticide use. For example, golden rice is enriched with vitamin A to combat malnutrition. Number 3 is evolutionary studies. Mutations provide insight into how species evolve and adapt over time. Number 1 is tracking evolution, and number 2 is antibiotic resistance. Genetic mutations serve as molecular clocks to study evolutionary relationships and trace common ancestors. For example, comparing humans and Neanderthal DNA to understand migration patterns. Number 2 is antibiotic resistance. Bacteria evolve rapidly due to random mutation that confers resistance to antibiotics. Scientists study this process to develop better treatment and control superbugs. For example, methicillin resistant Streptococcus aureus.